Hello all, I welcome you to another interesting session on Mendel's bicentenary celebrations. I am Mercy Rofina, a PhD student in Dr. Vinod Skaria's lab at CSIR IGIB. So we have with us here today Dr. Bibhas Kar, who is the head of the Department of Genetics and Molecular Medicine at Kokilaben Dhirubhai Ambani Hospitals, Mumbai. I must add that he has got over 25 years of research and professional experience in the field of human genetics, and his major areas of research interest are around clinical and molecular cytogenetics, molecular genetics, and genetic counseling. It's a pleasure to have you here in this platform with us here today, sir. Thanks for joining. And like I had mentioned, since we are here to discuss about the role of genetics, its importance, it would be great to start with what are your current research interests, or what exactly, is there a particular disease, or what exactly are you focusing on nowadays?
Thank you, Mercy, for your kind introduction. I'm grateful to you for, inviting and giving me this opportunity to speak on this very great occasion of Mendel's bicentenary celebrations being organized by CSR IGIB. Let me tell you very, frankly, I'm not an active researcher.
Okay.
But I have made some good contribution in clinical genetics, working over 2 decades on many different aspects of genetic diseases in Indian population.
Right.
I had my formal training in the field of human genetics while doing my PhD at Gujarat University, School of Science, Division of Human Genetics in Ahmedabad.
Okay.
Just I'll go little back to my career when I was doing PG in zoology. Those days we did not have any fancy subject what we are nowadays. We children are going through, or they are being trained. Those days we had only zoology, botany in biological science, and maybe life science.
Right.
So having the restricted facilities or the limited facilities, as I wanted to become a doctor, and we had a lot of restriction in our family structure because we are all 4 children. Okay. My father was a teacher, so it was difficult those days. And then I got into science. Okay. And I'm very fortunate to share with you that somehow my dream has fulfilled. Okay. In the sense, this is my explanation, in the sense that I'm associated with all the hospitals, premier hospitals, starting from my career. Right. So I'll just, tell you that the MSc time we had a specialization cytogenetics. There was a lot of demand. We have reproductive medicine genetics. Then we had insect, this entomology, and we had some few, specializations. And I opted those days, 86, 87, cytogenetics. Right. So that was available. So that is how my career started. And my research topic during PhD was cytogenetic studies in Fragile X syndrome in mentally retarded population in Western India. And the word mental retardations, now we cannot use it. It's no longer used because it is an offensive word. And instead, it is being referred as intellectual disability. Right. So as a clinical person, I wanted to say that because I should not be telling something wrong at this moment. So that is one aspect. My work was well appreciated in Indian Science Congress, and also there is an association in UK. It is called International Association of Scientific Study of Mental Deficiency. So I got quite fellowship and awards subsequently for my work. And then if I tell you the stories, which is very interesting, which I need to emphasize here, that I moved to from my PhD days, I came and joined at Sankar Netralaya. Okay. That is way back in 93 as a genetic scientist, and established the Department of Genetics and Molecular Biology from the scratch. Right. How difficult those days, but with very limited resources, with limited basic, we had a very basic facilities. And I started, engaging myself in various research projects in ophthalmic genetics.
Okay.
We had a project on blepharophimosis. We had project on retinitis pigmentosa. We had project on Oguchi disease, that is called night blindness. And definitely we could do very satisfactory work having our collaboration with the Otago University, New Zealand, and could publish our work in all very reputed journals, human molecular genetics and molecular mutation, human mutations and clinical genetics those days.
Okay.
So the cytogenetics, the blepharophimosis is called, this is a syndrome which is autosomal dominant, and cytogenetic and linkage analysis that is done previously implicated that BPS locus maps to chromosome 3q23 region in multiple cases of this syndrome. But our finding through linkage analysis suggested BPS locus maps to chromosome 7p, not in 3q. So that was a very interesting finding, 7p13 to p21. And this finding, as a geneticist, we can immediately, come to a conclusion. These findings provide a strong evidence that BPS is a heterogeneous, a genetically heterogeneous disorder.
Right.
Then I moved to, various projects, and we had actually very interesting work on Oguchi disease. As I said, it's a congenital night blindness, disease of congenital night blindness. There are different, diagnostic documents are there, how we come to a conclusion of a particular, disease in ophthalmic institutions like Sankarnathala, one of the premier organizations. So there also we actually found the association of this particular homozygous frame shift mutation that is arrestin gene.
Okay.
And in most, this particular family, I just wanted to highlight that parents, they had a heterozygous mutation, and all the children, they had a homozygous arrestin in this mutation, and they are all affected. Okay. So each and every cases that I have gone through, because my work is very diverse, I have been working on multifarious angle. As you know, I am in the hospital sector.
Right.
And my primary job, Mercy, is to establish new technique, do validations, and that should be available in our genetic test services, right, which will be useful for day-to-day, investigation and use in the clinical decision making. Right. So they are because in a hospital setup, we have to see what are the tests needed, what kind of demand you get from your clinicians. So my primary job is to establish, bring new technology, validate them, and then it should be available in our genetic test services, right, which can be used in day-to-day clinical practice. Right. I moved from, Sankarnathala, I moved to Apollo Hospital, Chennai. Okay. That is again way back in 1999, which is a nationally and internationally accredited JCI, NABL, CAP, and NABH, those accreditation process. And I got involved again mainly into diagnostic work, where I was working on multifarious aspect, where I established the facilities of clinical cytogenetics in the area of postnatal, prenatal, congenital, contiguous gene syndrome. We call it microdeletion syndrome, like Prader-Willi, this DiGeorge, etc. So those are another specialized area. And then slowly I developed cancer genetics. That is mostly the hematological disorder, screening all AML, CML, APML, multiple myeloma, myelodysplastic syndrome, etc. And then I also established slowly the solid tumor. We used to screen all, solid fees, tissue-based fees, and come to a conclusion, when we get a block from to do the HER2 new fees for breast cancer and ROS, ALK mutations, and Ewing tumor, various cancer and various such solid tumors.
Okay.
So after my, I'll just quickly go to my next move. And when I joined at Madras Medical Mission, I joined as the consultant and head of the department. I started from the scratch and developed a huge center called Center for Genetic Studies and Research.
Okay.
That is in 2011. So I'm just quickly moving to Madras Medical Mission from Apollo, where I involved both in diagnostic and research. I was very fortunate to do a little bit of research, which I was not able to do in corporate hospital like Apollo, because there we are very confined to taking care of the reporting on time.
Right.
Because turnaround time is very important.
Right.
So then I'm very fortunate to get involved in research. So my area of my research, again, as I said, I was working on cardiovascular disease, mainly on pediatric genetics. And I was also involved in reproductive medicine in the area of infertility and bed obstetric history, disorders of sexual developments. And we have a lot of publications, which you can always see in the literature. I will give you one very interesting case, as you rightly, as in the beginning of your introduction, you explained that you can discuss some of your interesting cases that you have come across. I had a patient family where there was this second degree consanguinity was there, uncle niece marriage, and they had actually 3 children, one after the other. They actually succumbed. They died due to the diagnosis. What was made is dilated cardiomyopathy. Okay. So now cardiomyopathy is a big entity. There are different types of cardiomyopathy. Dilated cardiomyopathy, restricted cardiomyopathy, and you have arrhythmogenic cardiomyopathy and hypertrophic cardiomyopathy. So the case that I am actually discussing is the dilated cardiomyopathy, where the one side of the ventricle, it will be dilated. Okay. And it will affect the other atria also, right atria. This is the left ventricle. It will affect the right atria. So the problem was, in this particular case, one after the other, they lost both the first child, male child, second child, male child, third child. They came to hospital and we did a lot of investigation. Echo was done. It was finally diagnosed, and then it was sent for genetic investigation. So when we started investigating, we did some preliminary work, and then we did the advanced molecular technique was used. That is the whole exome sequencing. And we could find this particular family, the gene that is we found is myosin binding protein, and that is a stop gain nonsense mutation, and involves 21 kilobase pairs of comprising 35 exons. Okay. So same thing, we could find parents to have the same genetic mutation, but in a heterozygous condition. But the child, this particular third child, which was investigated, was found to be homozygous, affected with this particular gene. So they were very upset. There is a big, huge battle going on in the family. Right. They used to come for, I'm just not going into scientific things now. I'm just talking about the patients and how we have to counsel, whether we could able to bring solace to their life, whether we can bring happiness to their life by providing a healthy baby was very challenging for me. Because from the day one, starting my career from Sankar Netralaya till the story that I'm actually narrating you, Madras Medical Mission, I'll come to Kokilaben later on. My primary aim and goal, and I was completely focused whatever we do, whether we work on retinitis pigmentosa or blepharophymesis or Oguchi or infertility or bad optic history or any syndromic cases like Downs, Patau or DiGeorge or Prader-Willi. My focus, my primary goal was to help the family and how to ameliorate the process in the population at large. Delineate or ameliorate this particular disease or defect which should not, or if to some extent can be checked so that it doesn't pass to the next generation. So in this particular family that I spoke about dilated cardiomyopathy, we called the patients, they were completely broken into pieces. I was personally, in touch with the patient. I was constantly calling them, trying to, be along with them at this difficult time. And finally, because time is a constraint for us, so I finally convinced them and told them, please go ahead and get pregnant. If you are planning to have a child, go ahead, plan for a child. And to tell you the very interesting news, she got pregnant, she conceived, it was very uneventful. She came, attended the clinic, sat for first counseling, again sat for second counseling, got convinced, continued the pregnancy, and we planned to do the advanced molecular test collecting the amniocentesis. I arranged collecting the probe, sent it to another organization, did the amniotic fluid test, genetic test, confirmed the test, found there is no mutation, asked the parent to go ahead, give birth, normal delivery, child is healthy, no mutation, and they are very happy couple. So this message, there are hundreds such, examples to give, but as we have time constraint, I may not go further, but the message that I want to give it to the young researcher, young geneticist, the people who are in the field of genetics or clinical genetics or bioinformatics or ecological genetics or different, clinical, medical, human, whichever areas they are working. I think the message is very clear.
Yes.
So anything that you wanted to ask me?
Okay, so sir, it was like a quick tour of your 20 years of your experience and how you had started and how you had got through the journey and what was your ultimate goal of choosing this field. So it was really very, very insightful, and the story which you had told us also, like how genomics or genetics is giving a ray of hope for patients and the kid, or either it be a prenatal or a neonatal. So that was really insightful, sir. This brings me to one more question. Like now there's like a more advancement in the genomic technologies or the genetic diagnosis when you compare it with few years ago, like when you started. So with all these advancements, it can be put together in a nicer way to shape our national health policies in a better way. So if you could share your opinions on what do you think is that a future advancement that is needed in this field so that we can offer a better quality of life for the patients, that would be like great to hear from you.
See, most important, we must understand how we are burdened with genetic diseases.
Right, sir.
We are completely burdened with genetic diseases. See, India is home to culturally and genetically a very diverse population are burdened with various genetic diseases.
Right.
And look at, due to high prevalence of recessive alleles owing to endogamous practices, rare diseases, they form a significant burden in India.
Right.
So you see, for an example, infants with congenital malformation, it's a huge burden. 4,95,000. I was just going through their paper, and with G6PD, it is around 3,90,000.
Right.
With beta thalassemia, the burden is 9,000 every year.
Right.
Down syndrome, we see 21,000. Coming to sickle cell anemia and other amino acid disorders, it is huge, around 15,000 to 20,000, 17, 16,000.
Right.
So I feel, see, why so much of burden? How to lessen the burden?
Right.
What are the pros and cons that we can evolve, or rather we can dictate, not dictate to propose.
Right.
That's what your question, right? The policy, what should be the policy?
What can be the changes?
I think the genetic knowledge for genetic, prevention is better than cure.
Right, sir.
So how to prevent? So health benefits will depend on how healthcare service providers ensures that genetic and genomic technologies are used effectively,
right,
efficiently, equitably, and responsibly for the population as a whole. So I feel genetic services and public health genetic programs that combine prevention as well as care for affected individuals and families. For an example, hemoglobinopathies,
right,
sickle cell anemia, thalassemia, and its variant carrier screening, premarital and preconceptional genetic counseling, and prenatal genetic testing. I again emphasize, these are very important, the services which needs to be provided, and this will be a critical making informed choices about marriages, reproduction, and lifestyle.
Right.
My second advice would be genetic services or genetic education. I think where patient comes to doctor.
Right.
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 I think this, very. This is, I'm seriously thinking that there should be in all medical curriculum. Medicine, it should be one of the very important subject, like anatomy. They study pathology, they study pharmacogenomics, they study forensic medicine. I think they should study first year, second year of MBBS. This is what my feeling.
Yeah.
They should study genetics seriously. Because I will tell you my day-to-day clinical practice, because I have seen there are doctors who are sending diabetes, they're sending for karyotyping. I'm sharing with you, but I cannot name where, who.
Yes, sir.
My approach to government, to policy maker, this should be involved in a level medical students who are the budding doctor, future doctor.
Right, sir.
Because they need to understand how genetic, what are the variation, how it is inherited. Even they don't have simple knowledge of recessive, how recessive disease is inherited, how dominant it inherited, what is the risk in recessive disease, how it is it a horizontal transmission, whether it is a vertical transmission, whether both male and female will be affected or not.
Right.
In X-linked, what happens? Why males are affected, why not female? So this basic concept in genetics, I think that should be in the curriculum. Right. And genetic service and public health genetic programs should provide simple, low-cost technology, cost-effective activities according to the prevalence and severity of the existing genetic diseases.
True, sir. True.
And my third aspect, or rather my proposal or my feelings, is genetic services rooted in primary health care. See, most of the times what happens, If knowledge is there in primary health care center with nurses, with
Right.
doctors,
Hmm.
they can only refer to secondary care hospital, and then it will come to tertiary for advanced genetic testing.
Correct.
But if that knowledge and know-how and is not available, I think there has to be some, kind of repeated, giving program in TV, radio, and giving some useful article, common for common public to understand in a very lucid, in a very simple language.
Right, sir.
Right. So such popular articles should appear. We should educate. We should call this, patient parents, the group, and make some small meetings.
Right.
So this way we can propagate. So this message should go.
Right.
So this is very important, that genetic service rooted in primary healthcare should be linked to regional, secondary, and tertiary level of care.
Right.
And prenatal genetic services, which is very, very common in North America and other, countries. It is the maximum screening being done in prenatal genetics testing.
Okay.
So these kind of services aimed at detecting genetic reproductive risk,
right, sir,
through carrier screening for prevalent recessive condition, because that is very prevalent in our population.
Right.
And prenatal screening for neural tube defect, chromosomal abnormalities, and so on and so forth. That should be established.
Right, right. So those were like really insightful points. Very unique also, like adding the genetics as one of the subjects for MBBS students, and also the point about spreading awareness in the patient groups, especially that is like one of the valid points, so that the science, whichever is being done, or the medical setting reaches more to the public. So that was really
one more things I want.
Yeah, sir. You can add.
One more things I wanted to tell you. See why I brought this, because the genetics is a massive science. It has a wide application.
True, sir.
Starting from medical to pharmaceutical to forensic to veterinary to agriculture. We are celebrating Mendel's bicentenary, event.
Yeah.
See how Mendel has created his contribution so vast.
Right.
You talk about agriculture, plant and horticulture, high yield crop variety. This is a green revolution.
Right, sir.
Talk about poultry, talk about fisheries, talk about dairy. There is a dairy, milk production, white revolution.
Right.
So this kind of revolution has happened, and this world of genetics is so vast. True. I think students, talk about forensic, DNA fingerprinting, paternity testing, organ transplantations, immigrations. Talk about pharmaceutical development of all vaccines, insulin, interferon, pharmacogenomics. So it's a vast course. True, and my message to youngster, all young budding scientists or young students who are interested to, go for taking a specialization to study genetics, and build up their career in the area of genetics. So the science is so vast. They can do specialization in human genetics, genomics, quantitative genetics, population genetics, classical genetics, quantitative genetics, ecological genetics. So they actually get employed, in they have a various opportunities to get in, have a very good employment in various biotechnology, health, and food industries and big sectors. Yeah, they can get job in academic research laboratories. Yes. So field is very exciting.
Okay. That was really a very insightful advice for all the young researchers who are about to start, or people who are already working. Like you have elaborately told about their opportunities which are in the field, and also that they'll have a direct impact in influencing or impacting the life of patients in terms of human genetics. by providing a better quality of life, either in a scientific setting or in a medical setting. So it was really a very interesting session we had with you, sir. You had shared your vast experience and also a very interesting story which we could connect, or any audience here who are going to see this video, they'll be able to connect it, and they'll be able to understand the importance of this field on this occasion. So thanks a lot for joining us today here, sir. It was indeed a very pleasure having you here in this platform and having this discussion with you. Thanks a lot, sir.
Thank you, Mercy. Thank you very much.
Thank you, sir.